Good morning, class, and welcome to week 9 of A&P2. We're getting there. We are over the hump and coming into the final few weeks here. So this week we're going to talk about the digestive system. The digestive system is also called the gastrointestinal system or the GI system. It's the system that's responsible for breaking down food that we eat, preparing it to be absorbed by cells, supplying the body with water, and then eliminating waste products. It includes the gastrointestinal tract and a number of accessory organs. The GI tract is a muscular tube extending from the mouth to the anus. It includes the mouth, the pharynx, the esophagus, the stomach, the small intestine, the large intestine, the rectum, and the anus. Accessory structures include salivary glands, and then the liver, the gallbladder, and the pancreas. They also include the teeth and the tongue. This is a nice picture that you're going to want to review, showing you each area. Like if we were to start up here, we've got the oral cavity, the teeth, and the tongue. And the purpose of those is mechanical processing of food, moistening food, mixing with salivary secretions. And then you can go down from there. So we are going to go over each of these things in detail. So I'm going to leave this for you to go over on your own. Our digestive system serves multiple functions in our body. So we're going to go through the 6 main functions. So the first one is ingestion. That is actually eating or drinking. So bringing materials into the digestive tract through the mouth. Then we have mechanical processing, which is also called mechanical digestion. And that is crushing or shearing. So what you're doing with your teeth, actually chewing your food, is crushing and shearing, and what that does is it makes materials easier to propel along the digestive tract. We also have a lot of mechanical digestion that occurs within the stomach and the small intestine, where food is getting worked upon by the muscles of the digestive tract and getting mixed and broken down. Third, we have chemical digestion, which is the chemical breakdown of food into organic fragments that can be absorbed by the digestive epithelium. In order for us to be able to utilize nutrients from the food that we eat, it needs to be broken down into its smallest parts so that it can be absorbed. So that's chemical digestion. Secretion is the release of water, acids, enzymes, buffers, and salts. These are mostly used to break down foods. Buffers are used to increase the pH, to offset some of the acids and protect our digestive epithelium. So secretion is done by the epithelium of the digestive tract and by our glandular organs. Absorption is the movement of organic substrates like our vitamins and minerals, electrolytes, their vitamins, and water through our digestive epithelium so that it can be, it goes into the interstitial fluid of the digestive tract, and from there it gets sent throughout the body for utilization. And last we have excretion, and excretion is also called defecation, and that's the removal of waste products from the body. So you intake the food, it gets processed through your entire digestive system, you absorb as much as you can out of it, and then you excrete the rest. This is a video. I can't tell you if it works or not, so best of luck on the video. Do make sure that you watch the crash course videos that are on our website though, because they are excellent as well. So we're going to dive into the histology of the digestive tract. In general, we contain 4 layers in the walls of the GI tract. The innermost layer is the mucosa. Under that is the submucosa. Outside of that is the muscularis externa. And then the outer layer is the serosa or the adventitia. The mucosa, which is the innermost layer, is a mucous membrane, and it's moistened with glandular secretions. So we have a lot of mucus production throughout our GI tract, and the purpose of that is twofold. One, it's to help with lubricating and moving bolus through our system, and two, it's for protecting the digestive epithelium from the acids that are produced within the system. Underneath the epithelium, we have a lamina propria, which is a layer of areolar connective tissue. We also have villi in the mucosa. So those are small projections that stick into the lumen. This is within the small intestine where we have these villi. And the villi increase the surface area for absorption of food. We'll look a little bit more at those in a little while. And then also within the mucosa, we have a layer called the muscularis mucosa. And the muscularis mucosa has two concentric layers of smooth muscle cells, and they can contract to alter the shape of the lumen and to move the villi. But don't confuse the muscularis mucosa with the muscularis externa, because we have some contractile function in the mucosa, and then we have additional smooth muscle layers in the actual muscularis externa. Submucosa is deep to the mucosa. It is dense, irregular connective tissue, and it contains our blood vessels, lymphatic vessels, and nerves that have to supply the mucosa. We have glands in the submucosa that secrete buffers and enzymes into the lumen of the digestive tract. So they secrete, and then their secretions go through ducts to end up on the mucosa within the lumen. The submucosal plexus is a nerve plexus that is sensory neurons, parasympathetic ganglionic neurons, and sympathetic postganglionic neurons. And all of that is part of the enteric nervous system or intrinsic regulation. So the enteric nervous system, if you remember from A&P 1, can function without the control of the central nervous system. However, it does also interact with them through sympathetic and parasympathetic fibers, but it does have the ability to function independently. The muscularis externa is dominated by smooth muscle cells, and we have two layers, and you'll find this throughout the digestive tract. So the inner layer is a circular layer, so it's encircling the small intestine or the large intestine. And then the outer layer is longitudinal, so instead of circling around the tube, it is going parallel to the tube. The myenteric plexus is within the muscularis externa, and you can remember M for myenteric, M for muscularis, so those two go together. Versus on the last page, we looked at the submucosal plexus, which is in the submucosa. So the myenteric plexus is nerves supplying its sympathetic and parasympathetic, which are also part of the intrinsic regulation of digestion. Parasympathetic nerves are going to increase muscle tone and activity. So remember with the rest and digest, we are increasing digestion with parasympathetic stimulation. With sympathetic stimulation, we're going to decrease muscle tone and decrease activity. So peristalsis, which is the movement of food through the digestive tract, is going to shut down with sympathetic activity, and it's going to increase with parasympathetic activity. And then our outer layer of the GI tract is called the serosa. So the serosa is a fibrous connective tissue, and it is the peritoneum. So we have the visceral peritoneum, which is a serous membrane that is on top of the organs, and then the parietal peritoneum, which is a serous membrane lining the wall of the abdominal cavity. So we've learned this in A&P1. It's very similar to the pleural membrane. So we have the visceral and parietal peritoneum. Serous folds in the visceral peritoneum become continuous with a sheet of membrane called the mesentery. And the mesentery is throughout your abdomen, and it's what holds your intestines to your abdominal wall and stabilizes the position of your organs. So we'll talk a little bit about that again in the next slide. The adventitia is a fibrous sheath covering the muscularis externa within just the oral cavity, the pharynx, the esophagus, and the rectum. So these layers do not have a serosa. They are retroperitoneal, so they're not covered by the peritoneal membrane. And instead of the peritoneal membrane, they have what's called an adventitia. So that's just a fibrous connective tissue sheath. So the mesenteries are made up of a double sheet of the peritoneal membrane. And there's areolar connective tissue between the surfaces, providing a route for blood vessels, nerves, and lymphatics to and from the digestive tract. And it stabilizes portions of those organs that it attaches to and keeps them from getting tangled up. You don't want your intestines to get tangled up because digestion will stop, you'll have an impaction, and that's just all terrible stuff. So the mesenteries, during fetal development, we have a ventral mesentery and a dorsal mesentery. The ventral mesentery mostly disappears, but it stays there in two parts. The first part is the lesser omentum up here, and the lesser omentum stabilizes the position of the stomach. So you see right here, the lesser omentum is stabilizing that stomach. And then the second part of the lesser omentum is the falciform ligament, and that stabilizes the position of the liver. So the lesser omentum all but disappears, the ventral mesentery disappears except for the lesser omentum and the falciform ligament. And then the dorsal mesentery turns into the mesentery proper, which you'll see right here. And the mesentery proper stabilizes the small intestine. So you can see all of these bits of the small intestine in there, and they all have their own sort of sheath that's holding them where they belong. And then the mesocolon, here's the sigmoid mesocolon, transverse mesocolon. So the mesocolon is all stabilizing the large intestine. And then the greater omentum is a large sac in the front that holds a lot of our fatty tissue. So here's another pretty picture for us. So you've got the falciform ligament up top. Maybe I'll do this. So falciform ligament up here, stabilizing the liver. And you can see the mesocolon in here that is attaching the transverse colon. All right, so I'll let you guys look through that, but there are a bunch of, it's all just double layers of peritoneal membrane that have sort of grown into the abdominal cavity and are stabilizing the positions of the internal organs. Okay, so now we're going to move on to how material actually moves through the digestive system. Peristalsis is active muscular contraction that propels a bolus. A bolus is what happens after you chew up your food and mix it with the salivary secretions. It turns into this sort of moist ball of mashed up food, and so that's a bolus, and that's what you swallow. So once you swallow your food, it is now a bolus. And so the muscular contractions push the bolus through the digestive system. So the first thing that happens is, well, this is just showing you that the bolus is going from the mouth toward the anus. And that's obviously the way that we want food to move through our system. So first, in step one, we have circular muscles. So remember we talked about the muscular layer that has a circular muscles that encircle the lumen, and then longitudinal muscles that are going lengthwise. So you can see this inner portion here are circular muscles. And if you see all the little circles there, it's showing them like that because it's cut in cross section. So the circular muscles, those are all wrapping around the lumen, and then the outer layer here is they're running lengthwise. So first we have circular muscles that contract behind the bolus. Second, these longitudinal muscles are going to contract and they're going to shorten the digestive tract. So they're kind of pulling the digestive tract up. And then we have in step three, this wave of peristaltic action. So the circular muscles are going to each contract in waves that will then push the bolus forward through the digestive system. Segmentation is contractions along the digestive system that are not meant to be propelling the bolus forward. Instead, they're contractions that are working on mechanical digestion. So they are mixing, churning, and breaking up that bolus into pieces where they can be utilized for absorption. So these are different ways that segmentation can occur, but it is important to know that segmentation does not propel the bolus forward, that it is really utilized for mechanical digestion. So a little review about the regulation of the nervous system. The alimentary canal, which is another term for the digestive system or the gastrointestinal system, has its own nerve supply, and that's the enteric nervous system. They make up the bulk of the intrinsic nerve plexuses, which we have already discussed, the submucosal plexus and the myenteric plexus. And this allows for local regulation of digestion. So you can have stimuli that's local, and that will create peristaltic action or release of hormones or release of digestive enzymes in that localized area of the digestive system. The enteric nervous system is also connected into the central nervous system, so it's under control of the parasympathetic and sympathetic nervous system. So remember, the parasympathetic is going to increase digestion, and the sympathetic is going to inhibit digestion. Fun fact for you, the enteric nervous system has as many neurons as the spinal cord and as many neurotransmitters as the brain. I find that to be absolutely incredible. Our digestive tract produces at least 18 different hormones. We're going to talk about some of them later in the lecture today. But these hormones are going to enhance or inhibit the neural commands. So they have mainly a localized effect, but they can have a more widespread effect as well. Other local mechanisms, prostaglandins, histamines, and other chemicals are released when chemoreceptors are affected. So they can affect just small regions of the GI tract for localized digestion and processing. And then we can also have local pH and local chemicals are affecting chemoreceptors. Physical stimuli are going to be stretch receptors. And so those are all monitoring local changes and can regulate on a local level. Sympathetic activity mediates secretion, motility, and relaxation. Sympathetic inhibits motility and causes vasoconstriction. So when you are in parasympathetic mode, you are digesting. When you are in sympathetic mode, you are shutting everything down so you can run away from danger. Neural reflexes are coordinated efforts to synchronize digestion in different portions of the GI tract. So as a bolus is moving down through the GI tract, things need to happen in a very specific order, and our nervous system is in charge of making sure that that all happens. We have a couple of different reflexes. The gastroenteric reflex stimulates motility and secretion along the small intestine. And the gastroileal reflex triggers the opening of the ileocecal valve, which allows your bolus to pass from the small intestine into the large intestine. By then, it's actually not a bolus anymore. The bolus is coming down from the mouth to the stomach, and then after the stomach, it turns into chyme, and then it sort of moves on from there and changes in consistency. This picture is showing us the neural and chemical regulation within the gastrointestinal tract. So we'll start up here, and what this is saying is that the central nervous system here is going to have an effect on the myenteric plexus. So remember, that's in the muscularis externa. And the myenteric plexus can then stimulate peristalsis and segmentation. That's going to move the bolus and break down the materials within the bolus. It can affect secretion, so secretory cells, so secreting mucus, secreting digestive enzymes. And it can affect enteroendocrine cells, and those are the cells that are going to release our hormones. When the enteroendocrine cells release hormones, those are going to travel back to the secretory cells. I'm sorry, I know this is getting really confusing, but so the enteroendocrine cells release hormones, and they can circle back and create secretion. Additionally, local factors such as pH, physical stimulation, that's going to induce stretch receptor activity. Chemical stimulation is going to induce chemoreceptor activity. And so those can directly create secretion, and they can also stimulate the enteroendocrine cells again, which can release hormones and then affect secretion. So we have multiple levels of control that can be localized here or more systemic from the central nervous system. All right, so we're going to start now with our tour of the gastrointestinal system. And our tour is going to start in the oral cavity. The mucosa within the mouth is stratified squamous epithelium. You can consider that that would be because there's a lot of abrasion and friction between the food coming into your mouth and the insides of your mouth as you are chewing and beginning mechanical digestion. Some other areas that we want to focus on, we have the hard palate. Oh, there we go. The hard palate up here, which is made up of the palatine process of the maxilla and the horizontal plate of the palatine bone. We have the soft palate behind that, which then extends into the uvula. The uvula is a little dangly thing at the back of your mouth. The uvula is what keeps you from swallowing too quickly, making sure that you have enough time to mechanically break down that food before you swallow it. We also have the tongue here, and let's see, I don't want to make this too busy at the same time. We have arches in the back of our mouth. The palatoglossal arch is one of them, and those two arches are on either side of the uvula. We have this region called the fosses, and that's the region between the uvula and the tongue right there. And the bolus is going to have to pass through the fosses in order to enter your oropharynx. What else do we want to know? The lips are in the front, and the area between the lips and the teeth is called the vestibule. I think that's all we need to really focus on this picture. Next we have our teeth. Teeth are accessory organs of digestion. With the teeth we have the enamel, which is on the outside. Here, the very outside of the teeth is the enamel, which is the hardest, let's see, let me find that. Enamel is the hardest biologically manufactured substance. So that's interesting to know. The teeth are within the alveolar sockets, right? The alveolar processes of the maxilla and the mandible. We've got the blood vessels and nerves that supply the teeth, and they enter through the apical oops, sorry, you can't see that. They enter through the apical foramen, and that's down here at the base of the tooth. Down toward the bottom of the tooth, we have the root canal. So that's the actual canal where in the root. So this is, here is sort of the delineation between the root of the tooth and the crown of the tooth. So the root is inside the bone. The crown is the part that's sticking out that we can see. So the root canal is this canal that the blood vessels and nerves are traveling through. The periodontal ligament is right up above that. And so that's the ligament that's actually stabilizing the tooth into the socket. In the root of the tooth, we have cement, which is the material that is keeping the tooth, keeping its shape. And then this whole region through here is the dentin. And the dentin makes up the bulk of the tooth. Let's see, what did I want to tell you about dentin? So dentin is the mineralized matrix, which is similar to bone. However, it does not contain cells. And then deep to the dentin, we have the pulp cavity. So the pulp cavity is the insides here. I am struggling, I'm sorry. All right, the pulp cavity is this inside that houses all the nerves and blood vessels. Back to the periodontal ligament. The periodontal ligament has collagen fibers that extend from the root of the tooth into the bone of the alveolar processes. So it's really securing that tooth into the bone so it's not falling out. The neck is the boundary between the crown and the root, and the gingival sulcus surrounds the neck of the tooth. And then cusps, these are cusps up here, and so they are projections off of the crown of the tooth on the what's called the occlusal surface. The occlusal surface is the surface where one tooth comes together with the tooth above or below. Development of teeth, we start with deciduous teeth, which are our baby teeth, and we have 20 deciduous teeth. I like to remind people of that when they are new parents and waiting for kids to start losing teeth, that whatever precedent you set with the tooth fairy, you need to hold it up 20 times. So that's always a good thing to keep in mind. We lose our baby teeth as we get older, and they are replaced by our adult teeth. And in the adults, we end up with 32 teeth overall. We have in each layer, so we have the top teeth and the bottom teeth. So we've got 4 incisors. So these front 4 teeth are the incisors. So we have 4 of those. And incisors are they're blade shaped and they're for clipping or cutting. So I remember that if you think of like you can bite off a piece of thread if you're sewing, and you use your incisors to do that. So they're for clipping and cutting. Next, we have our canines, and so also called our cuspids. So that's this tooth right here. Those are also called our eye teeth because they're situated underneath your eye. And those are for tearing and slashing movements. Then you have 1 cuspid on either side. Then you have 2 premolars, which are, you have your first premolar and your second premolar. That was a little far back. So it's these 2 teeth here are your premolars in there. And premolars are, they have flattened crowns and therefore crushing and mashing. Premolars have, when we're looking at the roots of teeth, you can have 1 root, 2 roots, or 3 roots. So our incisors have 1 root, our canines have 1 root, and our premolars have 1 or 2 roots. And then behind our premolars are our molars. We end up with 6 molars on top and 6 molars on bottom, so 3 on either side. So here's our molars, those last 3 teeth. And those are flattened on the top with 4 to 5 cusps, and they're for crushing and grinding. So premolars and molars serve about the same function, that crushing and grinding movement. Incisors are for clipping and cutting, and then the canines are for tearing and slashing, like breaking up meats and things like that. So our oral cavity serves multiple functions. Sensory analysis is tasting your food, determining all of the different characteristics of your food prior to swallowing. Mechanical processing is breakdown through the teeth, tongue, and surfaces of the palate. You use your tongue to sort of mash foods against your hard palate. Your teeth is tearing, slashing, crushing, and you're breaking it all down before you swallow it. Lubrication, you're mixing that food with mucus and salivary secretions, and that's going to lubricate it and make it easier to swallow. And then the beginning of digestion actually happens in the mouth. We begin with digesting carbohydrates with salivary amylase, and lipids with lingual lipase. And we'll talk more about those in a couple minutes. So once the bolus leaves your mouth, it goes through the pharynx, and we learned a lot about the pharynx last week, so we're not going to reiterate it this week. But the pharynx then dumps into the esophagus. The esophagus is a hollow muscular tube, and it descends posterior to the trachea. Last week we did pharynx, trachea, lungs. So this week we are looking at what happens when the epiglottis covers the glottis, cuts off the trachea, and you allow that bolus to slide past it and down into the esophagus. The esophagus is about 10 inches long. Don't memorize that. And it's just for to get a sort of picture of it, it goes from the C6 vertebrae down to the T7 vertebrae. It conveys solid food and liquid to the stomach, and the beginning of peristalsis happens here. We have two sphincters in the esophagus. The upper esophageal sphincter prevents air from entering the esophagus when you're breathing. You don't want a lot of air in your digestive system. So in general, that sphincter is closed unless you're eating, and then it will open and allow the bolus to pass. The lower esophageal sphincter prevents food in the stomach from re-entering the esophagus. That would be a reflux. So it is also contracted unless it is open to pass food, and that's preventing that acidic reflux from the stomach up into the esophagus. Here's a picture of our esophagus. And interestingly, the muscular layer of the esophagus is a little bit different than the rest of the digestive system. In general, the digestive system is all smooth muscle, but in the esophagus, you have to have some voluntary control in order to be able to swallow. So the top 1/3 of the muscular layer of the esophagus is skeletal muscle. The middle 1/3 is a mix of smooth and skeletal muscle. And then the lower 1/3 is all smooth muscle, and that's where our peristaltic action begins. Swallowing is also called deglutition. There's a voluntary phase and an involuntary phase. The voluntary phase would utilize the skeletal muscle. The involuntary phase would utilize the smooth muscle down at the bottom. And it proceeds automatically. So once food hits that, it's a swallowing reflex, which makes you start to swallow. It's divided into 3 phases: the buccal phase, which is voluntary, the pharyngeal phase, which is voluntary, and then the esophageal phase, which is involuntary. And that's that lower section of smooth muscle where peristalsis begins. So here's a little picture of the phases of swallowing. So step 1, you see that the bolus has just been, how the tongue presses up and pushes the bolus back toward the oropharynx. And this is what triggers the swallowing reflex. In step 2, begins at the palatal arches, and it's with elevation of the larynx that folds the epiglottis over the glottis to protect the trachea, and the uvula is going to come up and block off passage to the nasopharynx so you don't end up getting food or drinks up into your nose. That's very uncomfortable. And then step 3, so make sure you know that this is going this direction, okay? Step 3 is the esophageal phase, and that's where you have contraction of the pharyngeal muscles, forcing the bolus down into the esophagus. And at the bottom is where peristalsis occurs. Peristalsis is going to push the bolus into the stomach through the lower esophageal sphincter. All right, so that ends the esophagus, and once the bolus gets pushed through that lower esophageal sphincter, then the stomach takes over. So the major functions of the stomach is storage of ingested food, so everything you eat has to get dumped into your stomach. Mechanical breakdown of food, disruption of chemical bonds through chemical digestion. So we have mechanical digestion and chemical digestion happening in the stomach. And the stomach also produces intrinsic factor, which is that glycoprotein that's required for the absorption of vitamin B12. So if you have no intrinsic factor, you're going to be B12 deficient. Remember, B12 is derived from animal proteins, and so vegans and vegetarians often become B12 deficient. At the end of this mechanical and chemical digestion in the stomach, the end product is called chyme, which is CHYME. We're going to look at the anatomy of the stomach. We have 4 different regions. The cardia up here is, oh, sorry, that's the one I want. The cardia is the portion where the esophagus empties into the stomach. The fundus is this area up here where once the food comes in, the stomach actually comes back up and over the top above the lower esophageal sphincter, and so that area is called the fundus. Then we've got the body, and so the body is the main portion of the stomach. That's all body in between those 2 lines. And then we have the pylorus, and the pylorus is this end segment down here. A couple other things that I like to point out on the stomach. The stomach has 2 curves. It has a greater curvature and a lesser curvature. The lesser curvature is about 4 inches long, and the greater curvature is about 16 inches long. So it's pretty impressive. 16 inches is a decent size. So our stomach is a pretty good size, and it can actually hold a fair amount of material. We also want to point out the rugae, and rugae are these folds that you see inside the lining of the stomach, and those are what allow for the expansion of the stomach to hold a large meal. In the pylorus, we have the pyloric antrum, which is the very first part of the pylorus, and then the pyloric canal, which is this end part here that's going to go to the pyloric sphincter, and the pyloric sphincter is what allows the chyme to leave the stomach and enter the duodenum, which is the first section of the small intestine. Movement of food through the stomach, it's acted upon by mechanical digestion, which mechanical action is the mixing of food and gastric juices that are being secreted, and it helps to move the bolus, break it down and start to move it toward the antrum. The concentric muscles are working to break down those large particles, and this whole mixing and emptying can take several hours. So when you eat lunch, the food that you ate for breakfast is just starting to move into the small intestine. The rate of emptying of the stomach varies on the volume of food that you ate, the osmotic pressure of the food that you ate, and the chemical composition of the contents. The stomach wall here, we've got gastric pits, and so these are each gastric pits, which are this little involutions of the membrane. The gastric pits contain a lot of the cells that do the work of the stomach, the secretory cells. Beneath the gastric pits, we have the lamina propria, the submucosa, and then the muscular layer. The interesting part about the stomach that you would want to remember is that it has a 3rd layer of muscular tissue, which is called the oblique layer. So it's got your normal longitudinal layer on the outside, and then it's got your circular muscles in the middle, and then it has this oblique layer that just gives additional ability for mechanical digestion. This picture is a close-up of a gastric pit. We've got mucosal cells that are up in the neck of the pit here, so those are producing mucus. The mucus lines the walls of the stomach and protects the digestive epithelium from the highly acidic contents. The pH in the stomach is about 1.5 to 2, which is extremely acidic. So we need to protect that delicate epithelium. The parietal cells, which are here, are secreting that intrinsic factor that we talked about a second ago. They also secrete hydrochloric acid, and hydrochloric acid is used to break down our foods. The G cells, which are down here, secrete gastrin, and you can remember that gastrin, G cells, G gastrin. And so gastrin promotes increased stomach motility and stimulates the production of gastric acid. So G cells, gastrin, gastric acid. And then our chief cells, which are deep down toward the bottom, secrete pepsinogen. Pepsinogen is a proenzyme. Proenzymes are inactive. Once they get activated, they convert into an enzyme. So pepsinogen converts into pepsin, and pepsin is a proteolytic enzyme, which means that it breaks down protein. Our digestive juices contain mucus to protect the stomach lining, hydrochloric acid, which dissolves food, kills microorganisms, and stimulates that pepsinogen to be converted into pepsin, and then lots of digestive enzymes. Up in this mucus layer, the bicarbonate that is in the mucus is alkaline, and that is what protects us from that acidic contents. In the stomach, it's important that even though we have digestive enzymes, we're breaking down food, but no absorption of nutrients takes place in the stomach. All of the absorption of nutrients is small intestine, so the stomach is breaking down materials and then delivering them to the small intestine, but not actually absorbing anything. The small intestine is the main area for absorption of nutrients. That is the main thing you want to know. 90% of nutrient absorption occurs in the small intestine. Inside the intestine, we have what are called plica, and in this picture you can see all of these ridges. And here you're looking at this ridge here. So the plica are folds of the mucosa which slow the food passage and allows for greater absorption. The slower your food moves through your small intestine, the more ability you have to absorb all of the nutrients out of it. And then we have villi and microvilli. Villi are little folds, like small folds in the lining of the intestines. Microvilli, we've learned about before, are on each individual cell, increasing the surface area of the plasma membrane. So both of those together increase our absorptive surfaces. The small intestine has three sections. The duodenum, which is the first section, shown up here in blue. That is the mixing bowl. It receives chyme from the stomach and digestive secretions from the pancreas and the liver. So we need additional secretions to break down our food and make it able to be absorbed. And so the pancreas and the liver are delivering their secretions into the duodenum at an area called the duodenal ampulla. The duodenum is about 10 inches long. You don't have to memorize these, but I like to point that out because while the duodenum is 10 inches long, the jejunum is 8 feet long, and then the ileum is 11.5 feet long. So you have a lot of intestine stuck within your body. So the duodenum is the smallest portion. The jejunum is the middle segment. That's where most of the chemical digestion and nutrient absorption takes place, is within the jejunum. I would remember that. The ileum is the final segment, so it's the end, and it ends at what's called the ileocecal valve. And the ileocecal valve controls the flow of contents from the small intestine into the large intestine. Within the small intestine, we've got the villi. So you can see the villi here are projections of the mucous membrane. And so they have all of these epithelial cells running up and down that entire projection. And then on the cells themselves are the microvilli, which are increasing the surface area for absorption of the actual cells. Also seen here are the lacteals. And we talked about the lacteals last week. Those are large lymphatic vessels that collect lymph and can collect the larger structures that cannot diffuse into the capillaries. We have 5 different hormones that we're going to talk about that are regulating digestion. Gastrin is the first one. We've already sort of mentioned it, produced by the G cells, promotes stomach motility, and stimulates the production of gastric acids and enzymes. Secretin is released when the chyme enters the duodenum, and the primary function of secretin is to increase secretion of bile. The liver secretes bile, and it increases the secretion of buffers by the pancreas. So secretin is increasing secretions, so that should be easy to remember, but those secretions are bile and buffers. Gastric inhibitory peptide is secreted when fats and carbs enter the small intestine. Primary function is to inhibit gastric activity and stimulate the release of insulin at the pancreatic islets. Cholecystokinin, or CCK, is secreted when chyme arrives at the duodenum. It accelerates the production and secretion of digestive enzymes. It also stimulates gallbladder contraction, so more bile is going to get ejected into the duodenum. And then we have vasoactive intestinal peptide, which dilates regional capillaries, providing a greater ability to remove nutrients that are getting absorbed. More blood flow, more movement of nutrients. So vasoactive intestinal peptide is going to dilate capillaries so that they can uptake more of the nutrients that are being absorbed from our food. Digestion is regulated in 3 different phases. The cephalic phase begins before food enters your mouth. So when you see, smell, or then taste food, that begins the cephalic phase. The gastric phase is neural and hormonal action triggered by food reaching the stomach. In the intestinal phase, stimulatory or inhibitory actions are set in motion by food reaching the duodenum. So cephalic phase before you even eat, gastric phase in the stomach, intestinal phase when food reaches the duodenum. So we're going to look at each of those individually. So the cephalic phase begins with the central nervous system when you even think about food, or you see food, smell food, or begin to taste food. And what that does is it stimulates the central nervous system via the submucosal plexus to create secretion through these cells. So the mucus cells, the chief cells, the parietal cells, and the G cells all begin secreting before you even eat food. And what that does is it prepares the stomach to be ready to start digesting the food that is about to arrive. This can be altered by emotions. So if you're, some people are like stress eaters. So when you stress, this might stimulate your stomach. And then once your stomach gets stimulated, you're like, okay, I need to eat. This phase only lasts for a few minutes. It's because, once you smell the food, you're typically about to start eating. So this is a very short phase. That's the cephalic phase. The gastric phase is step 2. So this begins when food hits the stomach. It lasts about 3 to 4 hours. We've got stretch receptors and chemoreceptors that are getting stimulated by the distension of the stomach and the elevated pH through the gastric acids that are getting released. So those chemoreceptors are going to stimulate the submucosal and myenteric plexuses or plexi. And those are going to have the mixing waves. The myenteric plexus is going to create muscular contraction, and those contractions is going to start mixing up the contents of the stomach. And the submucosal plexus is going to create additional release of mucus, pepsinogen, all those acids and enzymes that are need to be produced to break down the bolus into chyme. And then our intestinal phase takes place when the chyme leaves the stomach and enters the duodenum. The enterogastric reflex inhibits contractions and allows for chemical digestion, and that is giving additional time for the breakdown of food. When lipids are present, it stimulates the release of CCK, cholecystokinin, and you remember that CCK is creating contraction of the gallbladder to produce additional bile into the duodenal ampulla, and bile is what we need in order to break down lipids, and so that is a great process right there. Chyme on the duodenum stimulates the pancreas to release its digestive enzymes and the liver and gallbladder to produce and release bile. Pancreatic enzymes and the bile enter the small intestine at the hepatopancreatic sphincter, which is also called the sphincter of Oddi. The small intestine secretes additional digestive enzymes like maltase, sucrase, and lactase, which are breaking down. Each enzyme has its own individual molecule that it's going to break down. Like lactase breaks down lactose, and lactose is the sugar that we find in milk. Enzymes and bile salts in the small intestine are continuing to break down the chyme that's been released from the stomach, and then nutrients eventually are absorbed and then sent to the liver for further metabolism. So that was all the gist of what happens in the small intestine. Once food leaves the small intestine, it enters the large intestine through the ileocecal valve. The large intestine is about 1.5 meters long, so that's 4.5 feet roughly. It's made up of the cecum and with the attached appendix, the colon, the rectum, and the anal canal. The colon is divided into the ascending colon, the transverse colon, and the descending colon. Our sphincters are the ileocecal valve, which we find right at the beginning, where the ileum is releasing contents into the cecum. And then the O'Beirne sphincter is toward the end. And what that does, I don't see it on here, but it would be right in this area, and that's going to release materials from the colon into the rectum to begin to stimulate defecation. So on here, we want to point out the cecum is first, the first portion of the large intestine. It's not very big, and it is accepting materials through the ileocecal valve from the ileum. Then we have the ascending colon, which goes up the right side of the abdomen, and the change, this 90-degree curve here, is called the colic flexure, the right colic flexure, or the hepatic flexure, because it takes place right underneath the liver. You can see how this is all situated over in that picture on the left-hand side. So the ascending colon goes up to the hepatic flexure, And then the hepatic flexure marks the boundary between the ascending colon and the transverse colon. The transverse colon proceeds transversely across your body over to the splenic flexure, or which is also called the left colic flexure. After the splenic flexure, then the descending colon proceeds down the left side of your body, and it then empties into the sigmoid colon, which is at the sigmoid flexure. And so the sigmoid colon is here. It's S-shaped. You can see that S that I just drew for the sigmoid colon. And then the sigmoid colon empties into the rectum. The tenia coli are 3 bands of longitudinal muscles that run the length of the colon, but they're not as long as the colon, so they're always pulling on it somewhat, and that is going to result in this gathered appearance. So make those internal ridges within the large intestine, and those ridges are called haustra. So that's here, haustra are those internal outpouchings within the large intestine created by the pull of the tenia coli. The tenia coli replaces the longitudinal, in the small intestine, the longitudinal muscle layer of the muscularis externa wraps all the way around the small intestine. In the large intestine, it does not wrap all the way around, but it is separated into 3 individual bands which run lengthwise along the outside of the large intestine. The mucosal surface is folded on the inside, called rugae, but there are no villi like the small intestine. All right, so the cecum is this first pouch here, receiving the chyme. The vermiform appendix is right off of that. Here's our appendix there, and the appendix, if you remember, has mostly a lymphatic function. The ileocecal valve is the sphincter that controls the movement of chyme from the ileum into the cecum. The O'Beirne, the rectosigmoid sphincter, oh, there. Okay, so that's going to be down here, is between the sigmoid colon and the rectum. And then the anal sphincter, there's actually 2 layers of the anal sphincter. There's an external anal sphincter, which is under voluntary control, and an internal anal sphincter, which is involuntary. Our layers within the colon, we have the muscularis externa, which are in the, we have the circular inner layer, but then also the longitudinal layer becomes the tenia coli. So you can see how that does not wrap all the way around the outside of the colon. We have simple columnar epithelium, goblet cells, which are mucus-producing cells, and we have intestinal crypts, which are similar to those gastric pits that go down into the epithelium, but you can see how the epithelial surface itself is flat in the large intestine. It doesn't have those villi that the small intestine has. At the end of the digestive system, we have the rectum and the anus. The rectum is really housing the materials prior to defecation. We have what are called anal columns, which you can see here. And then the internal anal sphincter is all of this tissue, and that is smooth muscle that is involuntary. And then the external anal sphincter is this outer muscle here, and that is skeletal muscle under voluntary control. And it's really awesome that we have voluntary control over our defecation, because you don't want to have to just defecate whenever it feels like, whenever you have that involuntary mechanism. Functions of the large intestine. The main function of the large intestine is reabsorption of water. There's very little nutrient absorption that occurs in the large intestine, but a lot of water reabsorption. That's very important. It compacts the contents into feces. So the more water it removes, the contents become more dry and packed together. Everything leaving the stomach is liquid. So from the stomach to the anus, all of that water needs to be reabsorbed, and it needs to come into a more firm package. We do absorb some vitamins that are produced by bacteria within the large intestine. So vitamin K, biotin, and vitamin B5 are all actually produced by healthy bacteria that lives in our digestive system. So those vitamins do get absorbed in the large intestine. And the large intestine is storing our fecal material prior to defecation. This water resorption, interestingly, we have 1,500 milliliters of material that enters the large intestine each day. And then through all of that reabsorption, we end up only excreting 200 milliliters of material. So we have 1,300 milliliters that get reabsorbed in the large intestine. The gastroileal and gastroenteric reflexes move the materials into the cecum from the ileum while you're eating. Movement from the cecum to the transverse colon is very slow, and so that movement through the large intestine is a very slow process, allowing multiple hours for water absorption and compaction of materials. The large intestine has what we call peristaltic waves. That is going to propel material through the large intestine. And then segmentation is happening within the large intestine. It's also called haustral churning in the large intestine because we have the haustra, and that is mixing the contents and exposing additional contents to the epithelia for anything that does need to be absorbed. Substances that are not absorbed in the small intestine pass into the large intestine. We have water and electrolytes get absorbed in the large intestine for usage by the body. As water is removed, the feces becomes more solid, and fecal matter is unused residue from ingested food, unabsorbed GI secretions, sloughed off epithelial cells, and intestinal bacteria. We have something called mass movements within the large intestine, which are those powerful peristaltic waves that move material through the large intestine. Those mass movements are unique to the large intestine. They do not occur anywhere else. So the large intestine is where mass movements occur. When we have solid waste products that have been formed through the large intestine, they enter the sigmoid colon and then the rectum to stimulate the defecation reflex. The internal anal sphincter is going to relax, and that's going to increase the pressure on the external anal sphincter. But the external is voluntary, so you can override that urge and wait for a more convenient time to utilize the bathroom. The reflex to defecate is triggered with a pressure of about 15 millimeters of mercury in the rectum, and you can override that easily. But if the pressure comes up to 55 millimeters of mercury, you can no longer override that sensation. So a lot of times with the stomach flu where you sort of lose control, it's because that material is moving so fast through your system and it's getting pressed so hard down into the rectum and anus that it overrides your ability to control the external anal sphincter. Here's a diagram of the defecation reflex. So it begins with distension of the rectum. When the feces moves from the sigmoid colon down into the rectum, it's going to distend the rectum. That distension is going to trigger stretch receptors, and those stretch receptors are going to trigger two things. First, they're going to stimulate the myenteric plexus to increase local peristalsis, and that increases the pressure on the anus. And then it's also going to stimulate parasympathetic neurons to stimulate somatic motor neurons and create involuntary contraction of the external anal sphincter, and that contraction is going to hold the contents until you are ready to defecate. So that is the main actual tubes of our digestive system, but there are multiple other things that we utilize that are accessory organs of digestion. So first we have our salivary glands. They are lubricating the foods that we eat and moistening the mouth, and also initiating carbohydrate and fat digestion. So the glands are the sublingual glands, which are down underneath the tongue, the submandibular glands, which are down sort of at the angle of the jaw, and then the parotid salivary glands, which are up sort of right under your zygomatic arch. Your salivary glands produce 1 to 1.5 liters of saliva each day. That's a lot of saliva. Saliva is made up of 99.4% water, and then also some mucus secretions and a few digestive enzymes. It contains bicarbonate, which helps. Bicarbonate, if you remember, is alkaline, so that sustains the saliva at a pH of about 7.4. That pH neutralizes some acids from bacteria, which helps prevent tooth decay and oral infections. We already looked over our three pairs. The submandibular gland produces about 70% of our saliva, and then the sublingual and parotid glands produce the other 30%. So the submandibular gland produces 70%. Main functions of saliva: lubricate the mouth, moisten the materials that are coming into the mouth, dissolve chemicals that stimulate the taste buds and provide sensory information, and then initiating the digestion of complex carbs with salivary amylase. We also produce what's called lingual lipase, and that begins the digestion of lipids. The liver is our next accessory organ of digestion. It is the largest visceral organ. That's probably important to know. Anytime something is the largest, that's an easy question. It's wrapped in a tough fibrous capsule. It's covered with visceral peritoneum, and it's divided into lobes. We have the left lobe and the right lobe. And as you can see, the right lobe is considerably larger than the left lobe. You're looking at an anterior view right now. The falciform ligament that we talked about earlier, which was part of the ventral mesentery, that stabilizes the position of the liver, is right here, holding on to the liver. And then posteriorly on the right lobe, it's posteromedial. So there are two additional lobes, one about here, let's see, I'll do that in a darker color, one about here and one about there, and they are the caudate lobe and the quadrate lobe. The liver is performing essential metabolic and synthetic functions in our body. Metabolically, the liver performs over 200 different functions. Main functions that we're going to discuss: carbohydrate metabolism, lipid metabolism, amino acid, which would be protein metabolism, removal of waste products, storage of vitamins and minerals, and inactivation of drugs. The main cells of the liver are called hepatocytes. They are capable of regeneration, which is excellent. If you damage your liver, it's important that you can regenerate it. Functions of the hepatocytes are to make and secrete bile. That's a main function. Bile is necessary for digestion, and so the hepatocytes are creating that bile. The liver, they also store glycogen, which helps to stabilize blood glucose levels. We learned about that back when we were doing endocrine in week 2 of this semester. Metabolizing fats and cholesterol, synthesizing albumin and globulin. Those are our plasma proteins. Remember when we did blood, we learned that the liver produces 90% of our plasma proteins. It also synthesizes urea, which gets excreted by the kidneys in urine. Synthesizes clotting factors, prothrombin and fibrinogen. Detoxifies chemicals, drugs, poisons, and steroid hormones. And stores vitamins and minerals for release as needed. In the liver, we also have Kupffer cells, which is the name for the macrophages that live in the liver. What they do is they filter out bacteria and contaminated blood from the intestines out of the portal system. And then they remove old blood cells and recycle the hemoglobin. The liver is divided into lobules by interlobule septums, and those are interlobule septa. So here's the interlobular septa that are separating these lobes. And then within the lobes, the hepatocytes are stacked up in plates, and those plates they look like the spokes of a wagon wheel. The sinusoids are capillaries that run between the hepatocytes, and they are receiving blood from the hepatic artery and the portal vein, and then that blood is getting filtered through the liver. Hepatocytes secrete our bile. Bile is a greenish yellow. I don't know who might have tasted it to know that it tastes bitter, because that seems gross, but apparently it's bitter tasting. It's an alkaline fluid containing bile salts, cholesterol, phospholipids, bilirubin, water, and electrolytes. The secretion of bile is called choleresis. What bile does is it aids in the mechanical processing in the stomach, creating large drops containing lipids. So it helps with breaking down fats. Pancreatic lipase is not lipid soluble, so pancreatic lipase can only interact with the surface of lipid droplets. I'm having a hard time with the word lipid today. So when you have fats in the intestine, in the duodenum, the pancreatic lipase works on the surface area of the fats to break them down. Bile salts, however, can attach to those fat droplets and break them down further in a process called emulsification. What that does is it breaks them down to expose an increased surface area for the pancreatic lipase to work and break them down even further. And that's going to create little tiny emulsion droplets that are coated with bile salts. And those are called micelles. M-I-C-E-L-L-E-S. So we know that our bile is created by the hepatocytes, but then we need to get it into the duodenum. So we have these things called bile canaliculi, and they look a bit like the canaliculi that we learned in compact bone. They are pockets that are between the hepatocytes within these plates in the lobules, and they collect the bile. So the bile is going to run into peripherally located bile ducts. So you can see a bile duct right here. The bile duct is going to collect the bile, and then they are going to join together into right and left hepatic ducts, and then those are going to join together to form the common hepatic duct, and the common hepatic duct is going to flow into the common bile duct. Excess bile is stored in the gallbladder, and it gets concentrated in the gallbladder until the sphincter of Oddi opens into the duodenum and allows for that bile to be expelled out into the duodenum. Blood supply of the liver. The liver has dual blood supply, much like the lungs did. So 1/3 of the blood supply is arterial blood from the hepatic artery. So that's oxygenated blood coming into the liver to supply the actual tissues of the liver. And then 2/3 of the blood is venous blood coming from the hepatic portal vein. And that originates at the esophagus, the stomach, the small intestine, the large intestine, and contains nutrients that have been absorbed. So we have venous blood that comes to the liver, bringing the nutrients that are absorbed out of the digestive system to the liver so they can get processed and utilized by the body. Those on that last slide, I meant to say that the all of the arteries and veins entering the liver converge and enter at an area called the porta hepatis. The gallbladder is a pear-shaped muscular sac which stores and concentrates our bile. It is located in the posterior surface of the right lobe of the liver, and we can see it here in green. Bile gets released within 30 minutes of eating when the gallbladder is forced to contract through stimulation by cholecystokinin or CCK that forces the bile out of the gallbladder through the cystic duct into the common bile duct. If the gallbladder concentrates too much, you can end up with gallstones. Gallstones can sometimes just pass through the duct and not be a big deal, but sometimes they get big enough that they can block off that bile duct, causing what's called a gallbladder attack, and then requiring a cholecystectomy, which is a surgical removal of the gallbladder. The pancreas is another accessory organ of digestion. It is retroperitoneal. It's made up of the head, which you see over here, the body here, and then the smaller tail down here. It's endocrine and exocrine. I believe that was an extra credit question on the midterm. So the endocrine cells of the pancreatic islets secrete insulin and glucagon, and we learned about that when we learned about the endocrine system. And the exocrine cells, which make up 99% of the secretions of the pancreas, are acinar cells and epithelial cells of the duct system, which secrete pancreatic juices, which are digestive enzymes. The pancreas is a compound tubulo-alveolar gland. There's a throwback to histology, week 3 of A&P1. The pancreatic acini are the blind pockets at the end of those alveolar glands. The pancreatic islets are areas of endocrine tissue that are sort of scattered throughout the exocrine tissue. So here's a pancreatic islet right there. And those are 1% of the pancreas have these endocrine pancreatic islets. You can see it over here that it looks different than the rest of this glandular tissue with the pancreatic acini. Pancreatic juice is alkaline, so it is made up of a lot of bicarbonate, which neutralizes the acidic contents in the duodenum. Remember, the chyme coming from the stomach into the duodenum has a pH of about 1.5 to 2, and we need to bring that pH back up. So pancreatic juices help to do that. And then they also contain digestive enzymes like pancreatic alpha amylase, which is going to break down carbohydrates. Pancreatic lipase is going to break down fats. Nucleases are going to break down nucleic acids, and proteolytic enzymes are going to break down proteins. Our pancreas is regulated through neural control as well as hormonal control. So the neural control would be through the vagus nerve, and the hormonal control is through secretin. So when the chyme is acidic and it enters the duodenum, it is stimulated to release secretin, and the secretin stimulates the pancreas to secrete its bicarbonate and fluids to help neutralize that acidic chyme. And the presence of fatty acids in the chyme stimulates the duodenum to release cholecystokinin, and cholecystokinin and acetylcholine released by the vagal nerve leads to the production of enzymes. All right, so we're going to do a quick overview of chemical digestion. There are 6 nutrients that we need in our body: carbohydrates, lipids, proteins, vitamins and minerals, and water. So we're going to look at how we already talked about water getting reabsorbed at the large intestine. So we're going to talk about carbohydrates, lipids, and proteins and how we break those down in our body. So you want to look at page 928 in your textbook. That has these beautiful pictures of how everything gets broken down, but we'll go over these for now. So the breakdown of carbohydrates, so you want to look at the top to see what is each picture discussing. So this picture is talking about carbohydrate breakdown, and it begins in the oral cavity with salivary amylase. Salivary amylase can continue to work for 1 to 2 hours until the food is down in the stomach, and then the increasing acidity of the stomach acids will eventually, what's the word I'm looking for? It'll eventually basically inactivate the salivary amylase. Then when we get down into the small intestine, the pancreas produces pancreatic alpha amylase, and what that does is, and the salivary amylase as well, they break down disaccharides and trisaccharides into monosaccharides. And monosaccharides are the smallest sugars that are able to be absorbed through the epithelium. So the monosaccharides are absorbed through the intestinal mucosa, comes through the brush border, through the cell body, and is diffused out into the capillaries, and then they are in the bloodstream and sent to where they are needed for processing. Lipids are similar. We produce lingual lipase in the mouth, and that works down into the small intestine. In the small intestine, we have bile salts as well as pancreatic lipase that are going to work to further break down those lipids into micelles. So there's that word micelles for you. And then the micelles can diffuse with monoglycerides and fatty acids, triglycerides and chylomicrons get created with diffusion through that intestinal mucosa, and then it's the chylomicrons that actually diffuse into our lacteals. They're too large to diffuse into capillaries, so they go into the lacteals, and then the lacteals are going to diffuse into the lymphatic system and be returned and entered into the systemic circulation at the vena cava. Last, we have proteins. Proteins are begin to be broken down in the stomach through pepsin. They get broken down into polypeptide chains, and then in the small intestine, those polypeptide chains are further broken down by trypsin, chymotrypsin, elastase, and carboxypeptidase. So those are all different enzymes within the small intestine that break our polypeptides into short polypeptide chains or individual amino acids. amino acids can diffuse through the intestinal mucosa and enter the capillaries, where they then get sent to the liver for processing. So this is showing us the amount of food and liquid that we consume with additional secretions by our body. So in any given day, we are going to consume about 2,000 milliliters of materials. We then secrete 1,500 milliliters of saliva, 1,500 milliliters of gastric secretion, 1,000 milliliters of bile, 1,000 milliliters of pancreatic juices, and 2,000 milliliters of secretions in the intestine. Now, starting in the small intestine, you can see that we are starting to reabsorb water and nutrients out of that material. And then in the colon, we have all of this water reabsorption taking place, so that we end up only eliminating 150 to 200 milliliters of material in the feces, even though we have had thousands of milliliters brought into our body and then secreted further into that digestive tube. And so I just want to point out here that we are so good at reabsorbing water and minerals and nutrients out of the food that we eat, that we excrete a very small amount in comparison to what we are taking in. Age-related changes in the GI tract are similar to other systems. We have a decline in the division of epithelial stem cells, which compromises our ability to repair and gives us greater susceptibility to damage. Our smooth muscle tone decreases, which leads to constipation. Remember, smooth muscles, the contraction of those are what create peristalsis and propel materials through our digestive system. So if peristalsis decreases, then constipation ensues. We have cumulative damage through which we recognize through loss of teeth. The liver becomes less efficient at processing toxins. We have an increased cancer rate because we have decreased immune function and decreased stem cell repair. So increased likelihood of cancer as we age. Dehydration becomes common because our osmoreceptors become less sensitive, so we absorb less water from from the bolus and the chyme and all of the things that are moving through the system. And the changes in the GI system have an impact, or changes in other systems also impact the GI tract. So our olfaction, our gustation, we smell less, we taste less, therefore we eat less. Reduction in bone density can affect the GI tract and can be affected by uptake in the GI tract. So at the end of each chapter, it shows how all of our systems are interrelated, and it's important that function overall decreases as we age. So that is our overview of the digestive system. Next week, we are moving on to metabolism and energetics. That is going to be a more challenging lecture. So enjoy this week. Enjoy learning about this system and how we move from eating to defecating and all of these incredible processes that take place along the way. And I will see you guys all next week.